This unit is going to cover basic media. Media can be basic and it can be more advanced, which we will get into in the later sections. What is called diagnostic media. It's used to actually differentiate bacteria and test to see what type of bacteria a person could possibly have been working with, discovered, and from there. So, what does media mean? It has nothing to do with cameras or pictures or anything like that. Media is what you work with in a micro lab and you grow microbes on, and then they become cultures. Auger plates. That's a staple of a micro lab. So, what is an auger plate? Well, auger comes in powder form, or you can buy it straight up as auger. We buy it in powder form. Excuse me for a second while I click around here. Sorry about that. There's an auger plate. See, there's the lid on the top, and then the auger is on the bottom. Of course, the lid comes off. What you do is you take the powder, you measure it, you put the right ratio of water, you boil the auger, you autoclave it, and then you let it cool, and then you pour it into plates, and you finally let it cool 100%. And all media that you start working with, if you're going to try to grow cultures, all media should be sterile. That means there's no living organisms. So this is a sterile auger plate. This is called a deep auger tube. This is a broth tube, and this is an auger slant. Now, and here's another picture. So the lid would be on top, and the auger is on the bottom. So when you work with micro, if you write instructions or if you write your name and your table number, you put it on the auger side, not the lid. The lid can come off. The lid can turn, move around. So if you're going to separate this into compartments or something, you do that on the auger side. See, here are the auger slants. What happens with those is you pour the auger, and before we get to that, so when you're storing auger plates in an incubator, you keep them lid side down. And the reason that is that condensation will form on the lids of the auger. And if you put that in an incubator, it'll turn to water droplets. It'll fall on your plate and it can mix things together. So you always store things lid side down. Auger slant, you see there, you pour auger into a tube, you set it at an angle, you let it cool, and it increases the surface area. Auger slants are good because if you're trying to keep microbes for a long period of time, they will last longer inside of a tube. So there's an auger slant. This is an auger tube, but it's called a deep auger tube. You can see it was not put at an angle, and so when it's solidified, it's just straight across. There's no surface area there compared to like this type. This is a broth tube. Broth is also a common medium that is used in a micro lab. Once again, to keep microbes for long periods of time. The only thing you can tell with broth is that you have growth, and broth will get cloudy. And I'm sorry to keep clicking around here. That says deep auger. So like I said, so here's a broth tube. I know I have another picture of one. There it is. So this one actually looks cloudy. Look, compared to the two of them. See, that one's kind of clear looking. I know it's got a tan color, but and then this one looks like it's got some growth in there. It's called turbidity. So this is what you would be working with in a microlab. Now I'm going to go back. So there's an auger plate. There's that picture I had up. There's a deep auger tube. It's not slanted. There's an auger slant. There's an auger plate. And there are the examples. See, there's a broth tube. There's a slant. And then this is an auger plate. We don't work very much with these deep auger tubes. We do a little bit. When you label things, you want to make sure things are labeled correctly. You want to put your name on them, your table number, the organism you're working with sometimes, the type of media it is. In other words, is it nutrient auger? Is it tryptic soy auger, TSA? You label all that. You want to be able to find your cultures when you start looking for them. And it tells you a little bit more how to label a test tube. And then when you discard anything, you put tubes into racks that are kept in bins. And then things like petri dishes, you put those into a biohazard bag, in which those are autoclaved. Yeah, you try to keep tubes in racks. And there was a video that showed that earlier, and I have a picture that we're talking about the introduction to the lab. You keep test tubes in racks. Okay, so the first thing you have to do what is called aseptic technique. And the first thing you want to do if you're going to work with some microbes, In lab, this is also a staple. This is called an inoculating loop. You see the loop end, and then there's the wire. Here's the handle end. Now, the way we do it at Howard College is when you're going to sterilize this, we use open flame, Bunsen burner, and we start here and work our way out to the loop. You're going to see a couple of videos where people actually just heat up the whole thing, and a couple of them are starting on the ends, but once it glows orange, it's sterile. And you always want to sterilize your loop when you're working with microbes because you don't want to contaminate what you're working with. And like it says there, when the wire is red hot, it is completely sterile. Now, transferring cultures, and you're going to want to read through this. There's a video, and it's the company Biorad that's in this video, where they are transferring cultures and they're doing aseptic technique. Aseptic technique is you are going to sterilize the loop. Okay, you're going to hold the tubes in a certain way and you're going to unscrew the cap in a way that does not touch skin. So right here, like where it's starting, if you're going to transfer something, if you have a culture means there's microbes growing in it. If you have a culture and you're going to transfer that to a sterile broth tube, you would sterilize your loop first. You would then pick up the test tube the culture was in. And I'm really trying not to go back and forth. So I have too many examples up here. So let's say this is a culture and you also have a sterile tube. First thing you're going to do is you're going to sterilize your loop like it says here. It says hold it like a pencil. You're going to unscrew the top of the tube. You would then heat the barrel of the tube. And you're doing that because this prevents contaminants from getting into the tube or bacteria from escaping. You're just trying to keep everything as sterile as possible. So you're only transferring the microbes you're working with. So you would heat the tube, and you can see the video for that. You would then take your loop. You would get some culture on your loop. You would take the loop out of the tube. You would then heat the tube barrel again and put the lid on it, and you're trying to minimize any contact with skin. You would then open up the sterile tube that you're going to transfer it into. You would heat the tube of that, and then you would put your loop inside if it's an auger slant, and I'm not trying to get too much in here. If it's an auger slant under number 13, you would put the loop at the bottom and zigzag all the way up to the top. Once again, there's a video about that. If it's a broth tube, you just kind of swirl it in. But once you have transferred it into there, you would then heat the barrel of the tube again. At the very end, you would then sterilize your inoculating loop. So that's called aseptic technique. Next, we are on to, this is a company called Biorad. We actually purchased their bioluminescent P-Glow kit. Here is an auger slant. This is actually just a needle, it's called. That you actually stab auger with microbes on the end. Here is a Bunsen burner. There's a lighter, and ours are similar to that. We have a canister, so this person's that controls how much oxygen is going to the flame. In essence, how strong do you want the flame, or how high up do you want it to go? So he's going to light it, and it looks like he did. I can barely see that. Okay, well, he's now adjusting how much air, slowly and dramatically. There it is. You have a nice little cone. See the blue cone inside? And I'm sort of watching this video with you as the first time, doing the John Madden commentary, and I would think the next move he is going to sterilize an inoculating loop. There he goes. Well, and he started with the loop end. We have always done it where you start with the end closest to the handle and then you work your way out to the loop. Now this is one example. I'm going to show you another video where you do this, but if you're going to transfer cultures, you're using what is called aseptic technique. So here he's heating the barrel of a bottle. He's holding the lid in his fingers, and now he inoculated his loop. He then heated the barrel again. Now he's placing the lid back on it because you're trying not to contaminate. Now he is streaking an auger plate. See the auger on the bottom, the lid on the top. And he should go back at this point after this, and he will then streak his auger slant to kill the microbes that he had picked up. he's basically spreading it around. And the reason you do aseptic technique is you're trying not to contaminate yourself or the culture. And what you're transferring, you want to transfer only the microbes you're working with. If you were to take that loop and set it on the table and then pick it up after you sterilized it, you'd have microbes in the table along with the culture you're trying to grow on this auger plate. And he is definitely spread. There he goes. And now he's then inoculating the slant afterwards. Okay, liquid to a liquid. Here we go. So he sterilized his loop. He is now going to hold that. He's going to heat the barrel, kill any contaminants that could possibly be on the rim and prevent that from escaping. He's now getting some liquid on the end of the inoculating loop. He's going to set that down. Now he's going to pick up another, and boy, pretty chrome test tube lid. We don't have that. Oh, it just slides off. Anyway, he's now heating that. And then next, he's going to take that loop and he's going to swirl it around. See, like that. He's going to heat the barrel again. He's then going to put the lid back on it. He's going to put that that test tube in the rack. Well, now he's going to go ahead and sterilize his loop. If you're going to transfer something to an auger plate, you don't really heat up auger plates. You're just going to either pick up the bottom of the lid, or you'll see in the video how they pick up the top, the lid, not the auger side, and they kind of do their work with that. All right, we're going to have a video here on, first of all, we have a person lighting a Bunsen burner. We do something similar to that, although that's a really good idea. We have matches. And she is now sterilizing some forceps. The blue flame is, of course, the gas mixing coming up. We have these in our lab as well. They're portable canisters. And she's sterilizing some forceps. And you see they're glowing bright red. So anything like that is sterilized. Dry heat and flaming is probably the most efficient way to sterilize something. The problem is that not many things can handle that type of temperature. And she's definitely got these sterile. Now, I'm waiting for her to pick up this auger slant on the bottom. And it looks like the length of this video, here we go. So here's an inoculating loop. There's the handle. Here's the loop end that you work with the microbes. And when you sterilize a loop, you start with the end that's close to the handle first, and you work your way out toward the end. Now she is basically just heating up the end of this bad boy. There we go. So see how she kind of had the handle in first? Anyway, that's how to sterilize a loop. This video is going to show how to transfer cultures between different media. Auger slants. And the heading on this video says from broth to broth, from broth to slant, and then from broth to plate, which is what we've done in our micro labs in the past. So let's see how she does this. Now she's explaining what she's going to do. And I'll probably just speed up and go right to the action. Okay, now we're going to light a Bunsen burner, and all these videos, they're using a little strike plate thing, which I'm starting to get jealous. Here's the the inoculating loop, and there is a broth culture. Now the difference between a culture and, well, there's a sterile broth, and then a culture has a microbe growing in it. So it looks like, here she goes. She's going to, sterilize her loop, and she just flamed the whole thing. Good deal. And man, that's a serious Bunsen burner she's got. So the first thing you're going to do is you're going to take the culture part, which is what she's doing there. Well, at least she is heating the barrels. Now she's doing it all at once, so she's doing this rather quick. So now she's taking the culture part, has a microbe in it, and then she's going to transfer that to a sterile broth tube. And next, she should heat, yep, there we go. We're going to heat the openings. And she is placing, wow, those are interesting lids, and then she's sterilizing her loop. So she's got at least good aseptic technique. She did that pretty quickly. Odds of contaminating anything are pretty low. All right, next, here we have an auger slant. You see it's sterile here. There's nothing growing in it. Here is a broth culture. She's going to sterilize her loop, and she does it very efficiently. Has a nice big Bunsen burner there. So if she did what she did before, she's then going to take the lids off of the tubes. Oop, sorry, had to add that in. And she's heating the barrel. You're trying to prevent contaminants from leaving or killing things there. Now she's taking the broth culture. This is very exciting. And next, she should streak from the bottom. And there she goes. See, she went all the way to the bottom, and now she's going to streak the entire slant all the way up. Good job. Heat the barrel again. And this has never stabbed the media. Nope, not unless it's intentional. And then she's going to now sterilize her loop. Now she is going to transfer something from a broth culture to a nutrient auger plate. And we use those as well. We also use something called tryptic soy auger. So it looks like here she's going to do the tube probably how we would do it. There's a couple of swipes. You don't want to break the glass, of course. Now she's going to take the culture out. She's going to heat the barrel, good aseptic technique. Put the lid on there. And then now she's picking up the plate. Now that's one way to do it. And see, she's streaking. And in this case, it looks like she's doing it's like a Z streak, but you can do that different ways. But the idea is you don't want to set the lid out on the table and leave the plate open for long periods of time because it will get contaminated. Okay, last thing in this section, streak for isolation. There are times where, and this is an example of one, and I'm just going to go through the whole procedure with you. You see these individual colonies? Sometimes you have what is called a mixed culture. Mixed culture means there's more than one organism growing in your culture, either a broth tube or another plate or an auger slant. And you're wanting to isolate something so you can see what you have, you do what is called a streak for isolation. It doesn't say it here. Anyway, mixed cultures have, yeah, there they are. Okay, will be mixed. Contain contain more than one organism. A pure culture only has one organism. So what you would do, this is the entire streak for isolation procedure. And there's a video for that as well under the media lab videos. It's the very last one on the bottom. A person does this. So what you would do, here's the drawing. You're going to have like a no streak zone. You try not to touch the sides of a plate. And so you're going to do this on top of the auger. You would sterilize your loop. You would then get some culture from a tube, and you would do the aseptic technique. You would heat the barrel. You would you put your loop in, get some culture on it, and a lot of people will actually number these on the back of the plate so you'll know which order you're going in. You can put a 1, a 2, a 3, a 4, and a 5 right here. But you're going to make a first streak with your loop. You're going to take your loop and do this with it. You can see the person, it looks like that was their first streak. So they took the loop and they just kind of went back and forth. That's streak 1. You're then going to heat the barrel of the tube. You're going to close it up again, your culture tube. You're never going to double dip, pretend you're at a party. You would then sterilize your loop, and then you would come over here and you'd make streaks that go into the first streak. And what you're trying to do is you're trying to spread it out where it's less dense so you can get individual cells, they'll grow into colonies. So you would make your streak number 2. You would then sterilize your loop again, come back and make streak number 3, and the whole time you're trying to make these streaks go into the previous streaks. You can see here, whoever this was, person did this, and then it looks like they did that. And then it looks like they had one of these here, and then they had some streaks like that. It didn't look like they went exactly in the order in our diagram, but that would be streak 3. Okay, you're then going to sterilize your loop again, and you're going to make a streak 4. Now, between 4 and 5, you do not sterilize the loop. And it's actually up here. So, yeah, do not flame between streaks 4 and 5. So, do not do that. And then the 5th one, you would do the 4th one here, and the 5th one, you kind of do that. And you can see they did something like this. And there you have individual colonies. That's a colony. This is not a bacteria cell. That's a colony. That's 1,000,000 cells. You can't see a bacteria cell. They're too small. So if you put a bacteria cell on Auger, they will grow into these colonies like this. And then what you can do is you can take one of those colonies and you can do another streak for isolation. And what that's called is a subculture. And it's a pure culture. You're only working with one organism. Because there you can see this is all the same color. There's not different colors or anything. And so you would then do another streak with that one to get individual colonies so you can work with that. All right. I hope this video helped. Thanks for joining me.
And that's the bottom line, because Stone Cold